Let's talk about the use of DNA fingerprinting in forensics. DNA fingerprinting. It's a chemical test that identifies the genetic makeup of an individual or other living organisms. It's used for a variety of purposes, the most common of which known from TV, the convicting of criminals. Apart from being used as evidence in court, DNA fingerprinting is also used to identify bodies, identify blood relatives, and look for cures for various diseases. DNA testing can also be used to determine if the remains are human or not. DNA fingerprinting is also known as DNA profiling, DNA typing, genetic fingerprinting, genotyping, or identity testing. The term DNA fingerprinting is misleading, and it does not only use fingerprints, but it can use anything that contains DNA, including hair and roots, blood, skin, tissues, skeletal bones or remains, teeth, saliva, semen, vaginal fluids, and urine. DNA is unique to each individual, and we are born with it. Some aspects are inherited, which is why it can be used to identify blood relatives, also known as familial matches. On average, 99.9% of our DNA is the same as every other human's, which leaves us with the 0.1% of DNA which displays our uniqueness. The only exception is an identical twin, which will have the same DNA.
1977, geneticist Sir Alec Jeffreys began researching human genetics using molecular biology. Sir Jeffreys is commonly credited with developing a DNA profiling process in 1984 while working in the Department of Genetics at the University of Leicester. However, the first patent that covers the process of DNA profiling, as we know it today, was actually filed a year earlier by Dr. Jeffrey Glassberg. This patent was based on his work done in 1981 while he was at Rockefeller University. Glassberg founded Life Codes Corporation together with two other medical doctors to bring his invention to market. In 1988, Jeffreys' career became successful as the DNA fingerprinting process that he developed together with Dave Werrett and Peter Gill of the Forensic Science Service was used to solve a criminal case of two teenagers who had been murdered and raped in the years of 1983 and 1986 in Narrabri. This was the first time DNA fingerprinting was used to successfully convict criminals. DNA profiling makes use of highly variable repetitive sequences. These are known as variable number tandem repeats, or VNTRs, with a specific variation of this known as short tandem repeats, STRs. VNTR loci are similar between individuals who are closely related, yet vary greatly, and it is highly unlikely for individuals who are not related to attain the same VNTRs. Over the years, various profiling processes have evolved, including DNA extraction, RFLP analysis, PCR analysis, STR analysis, AFLP, DNA family relationship analysis, Y chromosome analysis, and mitochondrial analysis. Various DNA databases have been developed over the years. One of the earliest applications of such a database was collected from 1996 to 1998 at the University of Cambridge. John L. Dawson and Kevin W. P. Miller compiled a mitochondrial DNA concordance which was prepared from data collected as part of Miller's PhD thesis. Nowadays, several DNA databases exist around the world. While some are private, the majority of the larger databases are controlled by governments. The United States maintains the largest DNA database, containing more than 13 million records as of May 2018. In the United Kingdom, it's roughly the same size despite the UK's population being smaller. And the rate of this database's growth and size are of growing concern to civil liberty groups, especially as police have wide-ranging powers to collect samples and may even retain them when a suspect is acquitted. Some highlights over the years with the development of DNA fingerprinting. 1985, the first DNA fingerprinting of a human was completed. This was when a mother arrived with her child in England from Ghana. Authorities suspected that the child was not the mother's and arrested her. DNA maternity testing was used to prove that the son was in fact the mother's child, and deportation was averted. In 1987, Nakamura invented the term VNTR. This describes individual loci where allele or possible gene forms are composed of tandem repeats which vary in the number of core units. 1990 was seen as the golden age of DNA research, and 20 years of research brought many new applications to light. 1995, a new DNA technique, which was PCR, was accepted into courts. PCR is a major step forward, both due to the ability to recover information from very small or very degraded samples and an increase in discriminating power. This was used in the case of the O.J. Simpson to help clear his name from murdering his ex-wife. In 2011, victims and criminals in the September 11 attack of the World Trade Center were identified through DNA identification of thousands of remains in the ruins. And in 2007 displays the largest DNA databases held by the United States of America, exceeding more than 13 million records. Forensic use of DNA fingerprinting is an extension of medical comparisons to detect mutations within patients to study disease-causing genes. These diagnostics are duplicated from clean tissue, and these samples require no knowledge of the general population. However, forensic DNA fingerprinting may not be reproduced and often includes samples that are degraded or contaminated with the possibility of being left by numerous unknown sources. DNA may be obtained from blood, dandruff, earwax, hair, mucus, saliva, semen, skin, sweat, tissue, urine, and vaginal or rectal cells, which can be found through either bite marks, used cigarettes, clothing, condoms, envelopes, fingernails, linen, tape, toothpicks, utensils, and weapons. However, DNA is prone to degrade when exposed to heat, humidity, and sunlight. Contamination beyond a crime scene may also contribute to inadequate samples of DNA. For example, in a laboratory setting with incorrect control settings, DNA may encounter contaminants such as bacteria, fungi, or yeast. Another obstacle faced is the requirement of an offender to leave sufficient quantity of blood, hair, or semen or soft tissue for a DNA sample. For example, in the case of a rapist, they often do not ejaculate due to being impotent. In addition, the offender is sterile. The semen could contain minimal amounts of DNA. However, polymerase chain reaction, or PCR, is a technique used mostly when the sample size of DNA is too small or degraded. PCR amplifies and replicates the amount of DNA from smaller samples to create larger samples of DNA, which may be used for sequencing, gel electrophoresis, or even cloned for further experimentation. The concentration of DNA needed for PCR is 0.1 to 1.0 nanograms. PCR initially requires Taq polymerase, which is a heat-tolerant bacteria. It uses this as an enzyme to replicate new DNA strands using present strands of DNA as templates. Primers such as single-strand DNA, which is approximately 20 nucleotides, are necessary for Taq to connect opposite strands of the DNA template into the PCR technique. With these ingredients, repeated cycles of heating and cooling to allow denaturation, annealing, extension for DNA replication. Denaturation is the addition of heat to approximately 96 degrees Celsius, which denatures the DNA strands, providing single-stranded templates. Annealing is the cooling of the reaction, which allows the primers to bind to their single-stranded DNA sequencing template. And extension is the addition of heat for polymerase to extend the primers for DNA sequencing. PCR is most commonly used over the RFLP method due to its fast analysis, minimal amount of DNA that is required. Even though DNA is degraded, PCR can still be performed. Its high power of discrimination, along with its high volume of DNA processing. PCR allows the analysis of STRs, which are the specific alleles in the VNTRs. The analysis of STRs proves to be a more accurate technique with the markers of smaller sizes, easier separation, and allows for clearer analysis of the DNA.
How DNA works. Think of DNA as a length of letters, A, C, G, and T. There are typically 6 billion letters in a human cell, and the DNA in all the cells in our body are the same. Each letter has a specific function, with some coding for protein, which can make the cell move or speed up in chemical reactions. Some letters do nothing, and others are known as spacer DNA. DNA is in the shape of a double helix, with letters paired up and facing each other. C matches G on one strand and A matches T on the other. The first step is the extraction of DNA. This process begins by collecting the DNA from its purest form from hair or hair roots, blood, skin, tissue, skeletal bones or remains, teeth, bodily fluids. Blood is normally the easiest, but DNA can be extracted from any cell or tissue. The sample is treated with a sequence of chemicals until pure DNA appears as a white solid. The next step is cutting. As DNA strands are very long, DNA that has been freshly extracted is quite sticky. The strands are also too long to be separated in the gel and need to be cut up. This is done using a restriction enzyme, which does not cut randomly but at specific letter sequences. This stage involves adding the restriction enzyme to the DNA using a pipette. This process takes a few hours. Separating. A gel is used to separate the different size of DNA fragments produced by the cutting. The blue dye is added to the DNA fragments, and blue DNA liquid is moved into the holes in the gel. The blue dye is only used to see where the DNA has been added to the gel to prevent adding two different DNA samples in the same hole. The gel is made from agarose, which is derived from seaweed and is simply a firm, pure jelly. The gel is placed in a colourless liquid, after which electrodes are attached and the power turned on. This results in the DNA fragments moving into the gel with the electrical current. As larger fragments move slowly than the smaller ones, the DNA fragments are separated. The gel is ready after a few hours and then checked with an ultraviolet light to make sure there is a strong DNA smear. The gel contains ether bromide, which sticks to the DNA fragments and allows it to be seen with an ultraviolet light. There are first steps to extract the DNA fragments. Next is today's extension on the end of the technique to produce a more detailed DNA profile. The steps that follow are done to make the DNA fingerprint visible so that it can be used to identify an individual by comparing its sample to one of the databases available, or in the case where a familiar match is sought, to another DNA sample. The next step is transferring. The DNA fragments separated by the gel is dunked in weak acid to convert them into single-stranded fragments. This exposes the DNA letters rather than them hiding in the middle of the double helix. Next, the DNA fragments are transferred to white nitrocellulose paper. This results in the paper carrying an exact replica of the DNA on the gel. This is known as Southern blotting, from the name of its inventor, Ed Southern from Oxford. The Southern blot equipment involves trays, paper towels and many solutions, so you can imagine it gets quite messy. Heavy books are also placed on top of the towels to press everything down, and the blot is typically left overnight. The next step is adding. The method described thus far has been a routine in labs since the 1970s. Alex Jeffrey's contribution to the process was the selection of the probe. The probe is a colourless radioactive liquid that controls which DNA fragments will be seen at the end of the process. It is a small piece of radioactive DNA that contains a very specific sequence of letters. The probe adheres to the DNA fragments that has been matching the sequence. It is very specific and will only stick to fragments with a matching sequence of letters and not to any other fragment. In DNA fingerprinting, the probe consists of a sequence of 33 letters that is present in repeating shutters of the genome, and only DNA fragments with these shutters are visible at the end of the process. They show up as dark bands we are all familiar with on a DNA fingerprint. To explain it differently, there are many DNA fragments in different sizes on the nitrocellulose paper. We then ask the paper to only show the fragments that contain a specific sequence of letters. The nitrocellulose paper and the probe are placed in a glass tube in a hybridization oven at 65 degrees Celsius for a few hours to allow the probe to convert the paper and to stick to the specific DNA fragments that contain the matching sequence. Afterward, the paper is rinsed to remove the excess radioactive probe liquid. The last of the process is the X-ray film. The nitrocellulose paper is placed against a piece of X-ray film in a dark room. The X-ray film records the radioactivity pattern of the paper, thus identifying where the probe has stuck. Once developed, the X-ray film will have the pattern of bands, which are the DNA fragments where the probe has stuck. The key to a DNA fingerprinting is the probe, the radioactivity part of the DNA that identifies many fragments that contain the mini-satellite repeats. These repeats contain the 33 letters of DNA that are used in the probe but repeated many times. The number of repeats is different for different people, resulting in DNA fragment sizes with different size repeats being unique to an individual.
STR-PCR technology is an excellent type of profiling for identification with blood, saliva and semen to help convict criminals, exonerate wrongly accused or identify victims of a crime, war or disaster. Some of these examples: paternity testing, a scene of a crime where these fluids have been left behind, a crash, a mass disaster to find a person's identity in things like an earthquake, floods, bushfires and terrorist attacks, rape victims, and identification of bodies in a homicide. At the scene of a crime, when fluids or hair have been left behind, they can be used for DNA profiling to incriminate or eliminate a suspect to identify a person that has been in the area recently. In a mass disaster, DNA from a body can be used to identify victims. 9/11 is a great example of this, where victim profiles were analysed against personal effects that relatives from around the world sent in, things such as hairbrushes or toothbrushes, to match DNA to identify those victims. Lineage markers are also well used in forensic DNA. Mitochondrial DNA and Y chromosome analysis are used in cases where there is a low proportion of nuclear DNA sample, or where there is a good sample from a female victim and only a small amount from the male perpetrator. For example, in a sexual assault incident where there is little or no sperm left behind. It can also be a good method to identify old skeletonized remains, or DNA from a hair shaft without a root attached. Mitochondrial DNA is found outside the cell's nucleus in the cell's cytoplasm. It is present in larger numbers than nuclear DNA. Mitochondrial DNA is less affected, however, for identification purposes than STIs, which uses nuclear DNA. This kind of profiling is good for identifying World War II victims or Vietnam soldiers by comparing DNA from distant maternal relatives with remains that have been recovered from historical sites. It can be used in mass disasters like 9/11 when the remains are highly degraded. Missing person cases where their highly decomposed tissue or bone are found, or the remains are highly charred. Bones that are found many years later. A good example of this was a mass grave near Yekaterinburg in Russia. Evidence from a mass grave was recovered and sent for DNA analysis. It was revealed that 2 individuals were recovered from a nearby second grave where children from the Romanov family, the last Russian monarchy. Future applications will be dependent on developing the current technology used. NGS, Next Generation Sequencing. Expanding the already useful data system and creating a quicker and more cost-effective one. Time is always critical in police investigations, as is the accuracy of the information which is produced. Forensics will one day be able to take a portable unit to a crime scene instead of taking the evidence back to the lab. NGS currently has high error rates though, which is preventing the system being used within the police forensics. The Haloplex Target Enrichment System and NGS analysis are currently being tested for law enforcement. The system has a quick 2-hour turnaround for analysis. It works on a hands-free basis, swab in, profile out. The system automatically does the extraction, amplification, separation, detection, and alert calling. LCN, low copy number, will also be a fantastic way forward for profiling. A fingerprint left at a crime scene that is unidentifiable will be able to be used to detect a complete DNA profile. Skin cells from a latent fingerprint will one day be used to identify DNA. DNA has come a long way since 1977. Over the years, it has helped to solve many crimes and put names to bodies or remains, even when just a tiny amount of tissue, blood, or skin cells have been left behind. As technology advances, samples are waiting in a climate-controlled storage environment. Tissue samples from the World Trade Center 9/11 attacks could not all be identified due to them being highly degraded. Hopefully one day in the future there will be major advances in technology that can give identification to all of these victims.